After studying this module, you shall be able to know which elements are present in our body and their average amount, classify the elements on the basis of their action in biological system, identify the dietary sources of essential elements necessary for our body, learn about various diseases due to deficiency and excess of different elements in our body.
In this module, we shall be discussing about the various elements present in our body which play an important role in our biological systems. These elements are classified into essential, non essential, trace, and toxic. Essential elements are absolutely essential, are necessary for life processes. Trace elements are also necessary for life processes. Non essential elements are not essentially. If they are absent, other elements may serve the same function. Toxic elements disturb the natural functions of the biological systems. Some of the trace elements like iron, copper, zinc, iodine, manganese, cobalt, etc., are also essential for life processes, whereas elements like lead, mercury, cadmium are toxic. We shall study some of these elements and their role in biological systems in detail. Let us begin with understanding the different elements present in our body. Consider the contents of the elements in the body of an average healthy person weighing 70 kilogram. It has been established that more than half of the total weight of an average man is accounted by oxygen. The amount of various elements in our body are oxygen, carbon, hydrogen, nitrogen, phosphorus, calcium, potassium, sodium, and lesser amounts of magnesium, iron, and zinc are shown. Let's say in our body, elements amounts of oxygen is 45.5 kilogram, carbon 12.6 kilogram, hydrogen 7.0 kilogram, nitrogen 2.10 kilogram, phosphorus 700 grams, calcium 1.05 kilogram, potassium 140 gram, sodium 105 gram, magnesium 35 gram, iron 4.2 gram, zinc 2.3 gram. You can notice that the content of rest of the metals does not exceed 1 gram, particularly copper, which is 0.11 gram, and manganese 0.02 gram. Therefore, only 2% of the human body is composed of metals, but much of the life depends on them.
Metal ions in the biological system. Metal ions, their excess and deficiency. Concentration of metal ions in our system is controlled within very fine limits. This control is generally exercised by certain biological complexing agents. The deficiency or excess of metal ions causes disorders, which leads to various diseases. This can be seen in figure. Calcium. Calcium is a critical element in all animals and human beings. A healthy human adult has about 1.05 kilogram calcium, of which 99% exist as phosphates, resembling the mineral hydroxyapatite in bones and teeth. The small remainder is in cellular fluids, existing in partly ionized and protein bound forms. The primary dietary source of calcium is milk, with smaller amounts derived from meat, fish, and eggs, and still less from non dairy foods such as nuts, fruits, beans, etc. Dietary deficiency of calcium is not a common problem in nations with high dairy production and protein intake, particularly since normal individuals can regulate intestinal absorption and renal conservation mechanism with great precision. Hence, human health problems related to geochemical distribution of calcium, its entry into the human food chain, and its bioavailability are relatively uncommon. Exceptions include very poor diets, such as those low in milk and animal proteins, or unusual physiological problems, other illness, such as intestinal malabsorption. In case of excess calcium, it comes into the blood as calcium is rejected by the cell and its salts are not soluble. So, excess of calcium leads to the formation of stones, hardening of arteries, and cataracts in the eye. Magnesium. Magnesium, an abundant element in the Earth's crust, is vital to both plant and animal life. Chlorophyll pigment in plants is a magnesium porphyrin complex. All enzymatic reactions in animals that are catalyzed by ATP, adenosine triphosphate, require magnesium as a cofactor. Oxidative phosphorylation, DNA transcription, RNA function, protein synthesis, and critical cell membrane functions are all dependent upon optimal magnesium concentrations. An average human being holds about 35 grams of magnesium, of which approximately 99% is either intracellular or it is present in bones. Of the 60% which is present in bones, two-thirds is a part of mineral lattice. Dietary sources high in magnesium include nuts, seafood, legumes, and vegetables. Meat has intermediate magnesium content. Potassium. An adult human has approximately 140 grams of potassium, of which more than 90% is both intracellular and exchangeable. Potassium plus is predominant cation in intracellular space. Since muscles contain most of the body's intracellular fluid, they also contain most of potassium. Since potassium is found in most animal and vegetable foods, dietary deficiency is exceedingly rare except under unusual conditions, such as diets containing high quantity of refined sugars, alcoholic individuals deriving most of their calories from low potassium alcoholic beverages in state of starvation, etc. Sodium. Sodium is the predominant extracellular cation in all animals, including human beings. An adult human body contains about 105 gram sodium, of which approximately 24% is located in bones and 65% is contained in extracellular fluid. Sodium ion equilibrium is maintained primarily by the kidney, the key organ in water and electrolyte balance. Sodium chloride is the predominant dietary source, although excessive dietary chlorine appears to have no significant ill effect on health. There is much evidence dictating that excessive sodium ion intake results in elevated blood pressure, hypertension. Cobalt. Cobalt is an essential element for humans, although it is present only in trace amounts. Only a little over 1 milligram cobalt is present in an adult human body. It is the main component of vitamin B12, cobalamin. Vitamin B12 is synthesized only by bacteria. It enters the human food chain from animal sources such as meat. The dietary deficiency of cobalt is likely only among strict vegetarians or when the intrinsic factor from the stomach that facilitates B12 absorption is absent or severely decreased, as is the case in pernicious anemia. Zinc. An adult has about 1.5 to 3.0 gram of zinc, and the largest amounts are present in liver and bone. There is evidence that zinc concentrations in blood and several tissues vary considerably in response to many stimuli. Zinc appears to be critical in many functions. Human zinc deficiency is inherited in infants and is termed as acrodermatitis enteropathica and is characterized by behavioral disturbances, diarrhea, hair loss, and severe periorificial skin rash, all of which respond with remarkable promptness to zinc administration. Similar syndromes have now been reported many times with penicillamine treatment of other disorders, presumably due to chelation of zinc, as well as during total parenteral nutrition when zinc was not added to the nutritional solutions for even as short a time as 2 weeks. A more chronic dietary deficiency of zinc combined with other deficiency include dwarfism, hypogonadism, and sexual immaturity, the latter devised with zinc therapy. There is much evidence for marginal dietary deficiency in humans. The effects include decreased acuity of taste, impotence, delayed wound healing, hypogonadism, and oligospermia, poor development, and possible fetal wasting and teratogenesis. Dietary sources range in zinc concentration from 1400 microgram per gram to 2 microgram per gram or less in fresh fruit and vegetables. Bioavailability of zinc is especially high from animal tissues and low from milk and from grains. The latter effect is apparently due to binding to phytic acid and fiber.
Let us now study about the molybdenum element and its role in biological system. The essentiality of the molybdenum in animals and human beings is assumed from its presence in the metalloenzymes like xanthine oxidase, and aldehyde oxidase. Molybdenum is also part of the enzyme sulfite oxidase, an inherited deficiency of which cause severe neurologic disorders and early death in humans. However, no naturally occurring molybdenum deficiency has ever been documented in animals or human beings, even though several animal deficiency have been produced experimentally, particularly by using the molybdenum antagonist. Molybdenum is present in very small quantities. Molybdenum appears to be readily absorbed from the gastrointestinal tracts and excreted primarily through the kidneys, though human studies are lacking in this regard. In tissues with higher concentration, such as bone, liver, and kidney, the molybdenum content can be varied with dietary intake. There is evidence that dietary molybdenum affects copper metabolism in animals and man. Higher molybdenum intakes causing mobilization and excretion of the copper. These effects can be elicited in man even with the naturally occurring dietary sources of molybdenum. Since molybdenum concentration in grains and vegetables varies enormously, and it varies from uh, 1 time to 500 times, and varies also with the soil content. The possibility of molybdenum induced copper deficiency in man is conceivable, through not reported. Now, moving to chromium element. The designation of chromium as an element essential to animals and human beings is quite recent. The major biological functions of chromium is an integral part of an organic complex, originally isolated from yeast, termed as glucose tolerance factor, we can say GTF. This complex apparently includes 1 in chromium 3 oxygen state and 2 nicotinic acid molecules, and may coordinate with 3 amino acid molecules, most probably glycine, cysteine, and glutamic acid. Experimental data indicates that glucose tolerance factor functions in conjunction with insulin and may in fact aid in binding insulin to sites of action. Other activities apparently include a lowering of serum cholesterol and triglycerides. An adult human has about 6 milligrams of chromium. Chromium in trivalent oxygen state is absorbed in the upper gastrointestinal tract, but only in very small amounts. But however, hexavalent chromium is better absorbed. However, the trivalent chromium is biologically active, which is an essential element. Trivalent chromium as glucose tolerance factor is apparently absorbed much better. Thus, conversion of glucose tolerance factor in the gastrointestinal tract may be important and may vary with age of the individual. Chromium in excess amounts can be quite toxic. Dependent upon the chemical species of chromium in 3-oxygen state is much less toxic than the hexavalent form of chromium. Chromium is a known carcinogen and toxic metal present abundantly in tannery effluents in India. As an estimate, about 80 to 90% of the tanneries use chromium as a tanning agent. Of this quantity, the hides take up only 50 to 70%, while the rest is discharged as an effluent. This rest amounts to nearly 75,000 tons per day. Today, the tannery industry, mushrooming in North India, has converted the holy Ganga river into a dumping ground. Several analysis reveals high concentration of chromium even in supposedly treated effluents. The residues can be traced even in crops cultivated with water taken from the river. The Department of Environment has identified the tannery industry as the biggest pollutant across the country. Electroplating can also release chromic acid spray and airborne chromium trioxide, both of which can result in direct damage to skin and lungs. Chromium dust has also been incriminated as a potential cause of lung cancer, and chromium has been shown to be mutagenic in microorganisms, causing infidelity, that is misreading during synthesis of DNA copies. Thus, the story of chromium once again illustrates the principle of essentiality in small amounts and potential toxicity in larger amounts. Now let's take the next element, that is the copper element. Normal adult human has about 100 to 110 milligrams of copper. Higher concentrations are found in liver, kidney, heart, and brain. The prototype functional deficiency of copper in humans is an X-linked inherited disorder called Menkes syndrome, that is kinky or stilly hair syndrome. Copper insufficiently was first suspected by analogy with the abnormal bull in copper deficient sheep. The defect appears to be decreased gastrointestinal absorption and cellular utilization of copper. The essentiality of copper is the consequence of its role in metalloenzymes involving several critical biochemical pathways. Several of these enzymes are noted here. For example, superoxide dismutase metabolizes the potentially damaging superoxide anion. Lysyl oxidase is a monoamine oxidase required for cross-linking Collagen and elastin, the structural macromolecules of connective tissues. Dopamine, beta hydroxylase, amine hydroxidase, tyrosinase are all copper containing enzymes that interconvert the major neurotransmitters dopamine, noradrenaline, and adrenaline, probably accounting for the high concentration of copper in the brain. The later enzymes, tyrosinase, is also a key step in pigment production. Cytochrome C oxidase is the key and terminal enzyme of the respiratory chain, accounting for more than 90% of the energy of muscular contraction. Ferrooxidase, which is better known as ceruloplasmin, before its role in mobilizing and oxidizing iron from storage sites are recognized, is believed to be account for 95% of serum copper and appears to be multifunctional protein serving as a major transport system for copper as well. Copper is widely distributed in food chain, a notable exception being cow's milk. Copper concentration ranges from 20 to 60 microgram per gram in animal tissues to less than 2 microgram per gram in leafy green vegetables and fruits to less than 0.2 microgram per gram in cow's milk, which is much less than in human milk. Infants, especially if premature and not breastfed, are therefore more susceptible to dietary deficiencies. Excess loss of copper from gastrointestinal tract due to diarrhea is the most common precipitating factor. In Wilson disease, copper concentration increases up to 100 times greater than normal. Copper is accumulated in a number of tissues, but in particular is found in the liver, brain, and kidney, which leads to liver and kidney failure and various neurological abnormalities. Death results if the condition is not recognized and treated. Now, the summary of this module. In this module, we have studied about the various elements present in our body which play an important role in our biological system. These elements are classified into four categories: essential, non-essential, trace, and toxic. Essential elements are absolutely essential, are necessary for life process. Trace elements are also necessary for life process. Non-essential elements are not essential. If they are absent, other elements may serve the same function. Toxic elements disturb the natural functions of the biological system. Some of the trace elements like iron, copper, zinc, iodine, manganese, cobalt, etc. are also essential for life process, whereas elements like lead, mercury, and cadmium are toxic. Note that no common element is toxic at levels normally encountered through almost anything can be harmful at very high concentration. Concentration of the metal ions in human being system is controlled within very fine limits. The deficiency or excess of metal ions cause disorder which leads to various diseases. So this module has made you aware of the various type of the elements associated with the biological systems and how the deficiency or excess of metal ions cause disorder which lead to various diseases.